Good afternoon, everyone. On behalf of Parul University, I welcome you all. Myself, Dr. Tanvi Dodia, Associate Professor and Head of Pharmacognosy Department, Parul Institute of Pharmacy and Research, Parul University. For today's session, we are honored to have with us Dr. Nikunjana Patel, Assistant Professor and Head of Department of Pharmacognosy, Faculty of Pharmacy, Ganpat University, Gujarat. Dr. Nikunjana has 15 years of teaching and 2 years of industrial experience. She has published more than 30 research papers, one book chapter, and presented more than 15 papers at national and international conferences. Being a recognized PG and PhD guide, she has guided more than 12 PG students. 2 PhD students have been awarded and 1 student is pursuing PhD under her guidance. Madam is working as State Coordinator, IPGA Women Forum. She is the Executive Committee member of Society of Pharmacognosy Gujarat chapter. She has received Career Award for Young Teachers for 3 years from AICTE. She has received grant for organizing national seminars from AICTE as well as Guj Coast. She is the life member of IPA, IPGA, APTI, MPA and ISTE, and she has been working as an associate editor in International Journal of Pharmacognosy and Chinese Medicine and also reviewer for many journals. Today, Madam will address on the topic standardization of Ayurvedic formulations, need of an hour for traditional medicine. Once again, I welcome you, Madam. Nikunjana,
Thank you, Tanvi.
Okay, over to you, ma'am.
Yes. Thank you very much, Tanvi and Parul University. I would like to thank all of you for giving me this opportunity and this platform to share my views. I'm going to start going to present my slide. Just let me know it is visible or not.
Yes, ma'am, it's visible.
Okay.
Okay, yes, visible.
Okay. So first of all, I would like to thank and very good afternoon all of you. The topic which today I am going to talk is a very known thing and very we can say the burning topic of pharmacognosy field, that is the standardization of Ayurvedic formulations, need of an hour for traditional medicine. We know very well about the Ayurveda. As a pharmacognosist and as a pharmacist, we know all the what is the Ayurveda. Ayurveda is the oldest healing system of the medicine. We experience the role of Ayurveda, particularly in this COVID situation. We have heard lots of about the immunoboosters and available some plants which are also used like the guduchi and all help into the COVID situation. Know about the Ayurveda that the word itself say the Ayu and the life. Ayurveda means knowledge of life. Knowledge of life. Ayurveda is actually not like the using of the herbs or using of some formulations to cure the disease. Actually, the Ayurveda is a whole science of life. That means how to live a healthy and long life. Using the herbs is one part of that Ayurveda, but other are there, some Dincharya, Riticharya, yoga, and all exercise, what to eat, what to not to eat, like that. All these parameters are there along with the use of herbs as a medicine in Ayurveda. So Ayurveda is not only the use of herbal medicine, not only the use of some formulations to cure the disease, to prevent the disease, but it is a wholesome, we can say the idea or the knowledge about the healthy and long life. Now, as a one part of Ayurveda, that is the formulations. So there are mainly two kinds of formulations, or we can say the two kinds of herbs are available in the Ayurveda. They are mentioned like single herbs and multiple herbs. So in a single herb formulation, it is not that much complicated. It is only a single herb, so standardization of that single herb is not that much problematic or difficult. but the multiple herbs is very difficult. So Ayurvedic formulations, if we see at a global level, so what is the role or what is there in a market about the Ayurvedic formulation? So if we look at this, graph, so this graph shows us about the prediction of the market, Ayurvedic market throughout the world from 2019 to 2016. Prediction is given. So approximately in 2018 it was 4.9 billion US dollar, and the prediction of the market is at the 15.5% annual growth ratio rate, it is going to be at 15.8 billion US dollar. That is almost 5 to 15, 3 times high within this 8 years. So right now we are at 2021. So we have already done half of this journey. So we can see that what is the ratio of increasing the market is 15.5 cumulative annual growth rate. So this shows us that how the Ayurvedic formulations are, we can say, the demanded worldwide. Every country wants to buy the Ayurvedic formulations. Even in India also, there is a huge demand of the Ayurvedic formulations. If we see the distribution or the Ayurvedic formulation by product wise, they are mainly as a drugs form. They are mainly sometimes as a cosmetic, or we can say the hair care products, on health care products and oral care products. If we see geographically, the North America region means it is divided into the 4 regions: Asia Pacific, Europe, America, and the rest of the world. But the demand all over the world is high. Just only the drawback, we can say, or the restriction related to the Ayurvedic formulation is only the standardization or uniformity, which we can say that. So Ayurvedic formulations, we know that they are prepared as per the classical texts, such as the Sarangdar Samhita and Vaisajya Ratnavali, and lots of more than 56 textbooks are there, classical textbooks, which are approved by our commission, Pharmacopoeia Commission, from which we can prepare the Ayurvedic formulation, as we all know that. Okay. Now, these formulations actually, which are written into these books or the classical textbooks, they are mainly designed and manufactured based on the unique principle of Ayurveda pharmacology, that is the Dravya Guna Shastra, which are very different from this modern pharmacology concept of the molecular medicine. We know that the network pharmacology or the multi-drug, multi-target mode of action in the Dravya Guna Shastra there means we know that the plant, which is having more than 100 or sometimes lots of chemical constituents are there. And if we talk about the multiple or we can say the multi-herb formulations, okay, so in that there is a very big complex structure of the chemicals, means a big complex mixture of the chemicals are there. But the Vaidyas or the Ayurveda Acharyas, they know the complexity of these chemicals, and they know this multi-drug, multi-chemical, multi-target mode of action of that particular drug, and that's why they have made a combination of some drugs. Okay, so this exerts, or we can say the Ayurveda formulations are mainly based on this network pharmacology or multi-drug, multi-target mode of action. Okay, and that is very different. We know that in modern pharmacology, or we can say the allopathic medicine, it is different, like lock and key type of hypothesis is there. One molecule, one target. Okay, single drug, single target type of concepts are there in a modern pharmacology. But this is a different in case of Ayurvedic formulations, and this is also one strength of Ayurveda because it works as a different targets, and it cure the disease from its root level, we can say that. Okay, but what happened? The market variations are observed, or marked variation, different variations are observed among the same formulations if we collected from the market, which are manufactured by different manufacturers. Okay, so say for example, if we collect the Triphala Churna from 3 different manufacturers, if we collect some other Chyawanprash from 3 different manufacturers, so we can see that there may be some difference, not very minute difference are always acceptable, because there are lots of variation into the raw materials also there. So this complexity of the herbal formulations is a major challenges in the Ayurveda drug industry, making standardization of Ayurvedic formulation a difficult task. So let's understand first things, the scenario of the formulations of Ayurvedic formulations. So if we talk about the past scenario, or we can say at the time of Charak Samhita, or if we go back 4,000 years back, then what was there at that time? The ready-made formulations was not there. So at that time, the medicines or the formulations were prepared by the physician themselves for their patients. So they are very much qualified. They are very much knowledgeable. They have idea. They can identify the plants. So, and they can modify. They are so much experienced that physicians modify the formulations, or they can make a formula themselves for a particular patient. Mean patient to patient, sometimes the formulation may different. Okay, so this is what actual scenario was there, and they were also trained in various processing, so they know how to process, how to make a vati, how to make a prasa, how to make a lepa, tela, everything they know how to make. The procedures are known to them. And even they know the different procedures of shodhana, means purifications of drug and everything. They were following the guidelines of the Shastra and experience that teachers. According to the immediate need of the patients, they were modifying the formulations as per their occupied understanding. As I told you, they are that much experienced that they can modify at a time of, we can say, the delivering the formulation to the patient. So this is actual scenario, means at that time not ready to use formulations are available. The Vaidyas go to the jungle or go to the garden where the plant is actually there. They collect them, dry them, or sometimes they do all the procedures and make themselves a formulation and give to the patients. So this is the actual scenario. And what is the present scenario? Present scenario, the physician, they are not involved at all into the manufacturing of drug. They are generally involved into the diagnosis and treatment only. And the drug manufacturing has gone to the pharmacist. Okay, so pharmacists are making the drug formulations, and in large scale production, we are right now what we are doing, we are making in a large scale production. So it is not like for a particular patient, it is like all the patients which is having the same type of symptoms they have. Okay, so if we make a herbal cough syrup, or if we make some Vasavleha, so Vasavleha is for all those patients, those who are suffering from cough, cold, or like such kind of symptoms they have. Okay, whether they are having age different, whether they are having a different situation, or we can say the different reason to having a cough and cold. Okay, but they all are having a Vasavleha, which is manufactured by the same company at the same time. They also compromise with the longer and lengthy procedures sometimes. Yes, in case of like Asava Arishtha, or in case of the Bhasmas, if you said if you have heard about the procedure of Bhasma, it is a very long and lengthy procedure. Sometimes in making the Vasavleha, or like in some other formulations. So for making a bulk production, means a large scale production, sometimes they decrease the time of trituration. Sometimes whatever the shortcuts they are using, so they compromise some procedure, and they took to the small time, or we can say small procedure. Equivalent substituents are added, yes, in the compound which are not validated, not available. Yes, sometimes what happen, due to the controversy or due to the unavailability of some plants, so the substitutes are also sometimes added in place of that particular plant. So this is also happen, and the exact important procedures either altered or made shortcut, as I told you. So this is what the difference between the past scenario, means actual what the Ayurveda is there, actual how the Ayurvedic formulations were prepared, or they must should be prepared. They should be prepared right now also, but due to the large scale population, due to this modern scenario, the companies are making in a bulk production. So this is one of the parameter which affect the standardization or the quality of, actually we can say, the quality of Ayurvedic formulations. So this led to the decline in the quality of Ayurvedic drug. Yes, of course. And so standardization of Ayurvedic drug is thus felt necessary. Okay. So let's understand by some more examples. Here I have taken some example from the published data from some articles. So this one is taken from the one article in which that the publisher or the scientist have taken 3 different brands from the market of the Triphala Churna, and you can see just only the morphological appearance. The color is also different, like the brand A is having a brown color, brand B is having a light brown color, and brand C is having a yellow brown color. So starting from morphology, it is different. You can see the variation. Even taste also having a variation. The brand A is having a salty and sour taste, while the brand C is bitter in a taste. So you can see how the variations occur, or the variations are there in the marketed formulations. The same brand, they are also tested for physicochemical or properties like water soluble extractive value. You can see that no doubt they pass the standards of the pharmacopoeia, like not less than 35%, but you can see this is a 50% and here it is a 40%. So this is also a difference. Actually it passed the pharmacopoeia limit, so it is a valid thing, but among them also the difference is there. Here you can see in case of the Vaiswana Churna, they have done the TLC. Mainly it contains the Terminalia cebula, Ami, Apium leptophyllum, Gingiber officinalis, and rock salt. So this one, T1, T2 and T3. T1, T2 are the marker one. T3 is the Vaiswana Churna formulation formulation I, then D and S. These are the 3 Vaiswana Churna formulations are there. Okay, so here you can see the number of spot. In case of T3, you can see this is very small. 1, 2, 3 and 4 are there. These are absent in case of T4 and T5. The same thing is also over here. You can see this is not present into this. Okay, so this is how the variations are occurring in the marketed formulation. The same Vaiswana Churna, if you look into this table, water soluble extractive value and alcohol soluble extractive value in the formulation I, S and D. So majority here you can see this is a 14% alcohol soluble extractive value, and this is 28%, almost nearby 20, means double. You can see this is 14%, this is 14%, and this is 28%. So almost double the alcohol soluble extractive value in case of the formulation I, and it is half in case of the formulation D. So how it is possible? Okay, same way here 44%, 38%, and this is 50%. So these are some examples by which you can see that in case of Trikatu Churna, water soluble extractive value and alcohol soluble extractive value, this is 0.15 to 2.32, means almost it is double or more, not double, more than that. Okay, here also 0.56 to 1.4, this is 3 times higher than this one, marketed formulation 2. So these are some examples. Even more are there. Hingvastak Churna, again this is the difference, 7 and this is 14, nearby 13. So it is almost double. Okay, here it is also, this is 2 and 3.3. So again in case of the Draxarista, you can see the matching of this TLC fingerprinting, the spot, this one darker one spot, these are 11, 12 and 13 are the 3 different formulations, and this one is the extract of this plant, and you can see the absence of this marker compound, or we can see these 2 compounds, 1 and 2, into this last 2 track number 12 and track number 13. These are all 1 to 10 are all about the raw materials which are used for the making of Draxarista. So here also you can see that the variation into the chemical composition of this Draxarista available into the marketed formulations. So by seeing this all example, by viewing this all example, we can conclude or we can say that there is a need of standardization of Ayurvedic formulation. Yes, of course, it is required because it is not worldwide accepted due to some reason. Means the reason we'll see later on what are the reason, or we can say what are the parameters that we can control for the quality of the Ayurvedic formulations. So before that, we should know that what do you mean by standardization, means what is the standardization, that first of all we have to understand. Okay, so standardization is actually the process of evaluation, a process of evaluating particularly the quality and purity of crude drugs by implies the various parameters like morphological, microscopical, chemical, physical and biological observation, which is termed as a standardization. Okay, so it is the process of evaluating all these parameters like morphology, microscopy, chemical, physical into the crude drug. So this is called a crude drug standardization. Okay, so the same thing if you apply onto the formulations, so it includes not only the raw material standardization, it also includes the process parameters. Okay, if we shorten down this definition, the standardization is a numerical value or a specific property that quantifies the purity and quality of drugs and formulations. If further we shorten down the definition, or in other words, the standardization of the drug means the confirmation of its identity and determination of its quality and purity is known as a standardization. Very simple but a very meaningful definition is that the confirmation of its identity and determination of its quality and purity by the chemical, physical, biological observation, that is the thing that we should know that. Now, standardization of Ayurvedic formulation. If we talk particularly about the Ayurvedic formulations, the standardization of the Ayurvedic formulation deals with the ensuring the establishment of the standards for the quality and purity of raw material. Very first thing is the establishment of the standards for quality and purity of the raw materials. Quality control during the drug manufacturing process. This is very important. Then production of a good quality finished product, storage and distribution to maintain the quality of final product. So this is actual what the standardization of formulation. So standardization of formulation is not only the checking the raw material quality, not only checking the finished product quality, it is starting from raw material to the storage and distribution until that, to making the insurance to ensure that the quality and purity of that, the whole formulations remain the same throughout the until its expiry date. So this is called the standardization of Ayurvedic formulation. Okay, now standardization, if we talk about the standardization, it is not a new things, not a new word, not a new agenda. It is actually there in Ayurveda. Okay, it is mentioned. In Ayurveda, standardization has been well defined and documented in the classical as well as the contemporary texts. But this have been written with an individualistic intent and not for the industrial or commercial purpose. So this is the difference. It is there. It is there in Ayurveda in the textbook, the standardization of the formulation or what is standardization, what is quality, what is purity, how to maintain, what to do the maintaining of this quality. Everything is there, but it is individualistic. Means, as I told you, the formulations were prepared for individual patient, not for all other patient or all the people in a group of, we can say in a group of people. Okay, so it is there, but it is for a particular patient or for particular formulations. Okay, it is well explained with the protocol in Charak Sanhita, particularly in the chapter of the Bhaisaj Pariksha, means how to do Pariksha, means examination of the plant or raw material or the formulations. It incorporates the principle of standardization starting from the cultivation of the raw material drug to its uses by the individual patient, covering the manufacturing process, storage conditions and method of the use. Okay, so starting from cultivation means right now we know that good agriculture practice is there, good cultivation practice is there, then good storage and then good laboratory practice, GLP is there, good manufacturing practice is there. So everything is mentioned over there, but in a different words they have there. So if we look about the ancient protocol of the drug standardization, what was there in Vaisajya Pariksha, it was there like idameva prakritim. Idameva prakritim means nature of drug. So they have written the nature of the drug. It's a guna, it's vipaka, all everything. The nature of drug was also written over there. And then even gunam, specific properties of that drug. Okay, about the specific properties, like their leaf drugs are there, what kind of ushna properties it has, or like guna is there. So everything was written over there. Even prabhavam, specific effect of the drug on a particular patient, particular human being, is also written. Okay, the asmin deshe jatam means from where it was cultivated, from where it was found, from where it was picked up. Okay, so that is also written into that. Same way, collected in a specific season means time of collection is everything is mentioned. We see about each and everything with example. So if we look about the things of cultivation, the place of cultivation and collection of raw material, that should be also written, or that was there in a Ayurveda, or that was there in ancient text, that the quality of the soil and the nature of the place of cultivation, that you must have to check first before collecting that drug. The state of the drug, where it was collected or procured, yes, it is fresh taken, or it is taken as a dry form. Okay, so everything, whatever mentioned, you have to check the state of the drug when it was collected or procured, means age of that plant, the if it is collected, ripened, if fruits are there. Okay, so everything is there. Then richness of nutritional or the medicinal principle like guna, virya are vital, so that you have to check. There is a clear identification of the areas or the soil which are not fit for the medicinal or consumption purpose, like cremation ground. You never have to collect the drug from this nearby the cremation ground. Religious places, nearby the big trees. Okay, you not have to collect the drug from near to the big trees, like the people, or like we can say the Ficus bengalensis as Ficus religiosa, or near the roads and the passages. Okay, so this is the place from where we do not have to collect the drug. Say for example, one example is given over here of the Albizia lebec, that is the shirish. We know very well the bark of the shirish used in various formulations. So what they want to say here in this example, the bark of the shirish is mainly used for the preparation of powder and grita formulation. But if you want to prepare arista, asava arista, then instead of taking the bark, we have to select the inner portion, that is the heartwood. Okay, sar bhag means heartwood is taken. And even some of the scientists have also tested the pharmaceutical, analytical and pharmacological, even in the clinical evaluation of this formulation. One asava is prepared from the bark and one asava is prepared from the wood. Then they have also checked the efficacy, and they have found that the asavam, which is prepared from the heartwood, is showing good clinical parameters, even good all other pharmacological and analytical parameters compared to the asavam prepared from the bark of the shirisha. Okay, so that is also have to always in the consideration. The time of collection, yes, of course, it is very important. We have to always check, or the time of collection are also written into the ancient textbook also. So we should have to follow that. If we see the example of the Rauwolfia serpentina leaves, they contains maximum alkaloids like 0.5% in the month of July, August, means a rainy season, and they are very least or a lowest 0.1% in the hot season, like in May, June. So whenever we want to collect, then we have to select always a rainy season. Okay, the same thing is also happen in case of the Vasaka. Okay, so the Vasaka have throughout the year different components, means the different percentage of alkaloids, and maximum percentage in the month of August, September and October. So we have to collect the Vasaka mainly in the August, September and October. Then the storage conditions. The definitive guidelines are given for the collection and storage of certain drugs in the ancient textbook. Say for example, Madan Phala, if we talk about that, that is known as dal. in Gujarati. Good quality of the fruits are collected and placed into the bag of kusha, covered in the cow dungs and placed in a husk for 8 days. And then the whole procedure is given, like seeds which are rich, they are separated, they are placed in a ghee, then curd, then honey and til kalka, and then dried, and then they are kept into the earthen pot which is covered, and the pot is suspended from the roof. Okay, even they have some protocols or some instructions are there written into the textbook that the Vaisajya Shala, means this manufacturing unit, should be constructed at the place near a natural source of pure water, beyond the possible place of the contamination, such as the cremation ground or the public places or the toilets or religious places. Okay, and only the qualified and healthy workers are only allowed to work into this premises, Vaisajya Shala. So these are all our Schedule T, or we can say the GMP and everything what we are talking right now. That is all written into our textbook. The processing techniques, yes, of course, it was also mentioned into that, but drug to drug, the processing techniques was different. Say, for example, if we're talking about the Sodhana of the Nux vomica. Nux vomica, we know it is a very toxic drug. These seeds are toxic. So the crude form Nux vomica contains strychnine 1.92% and brucine 0.93%. But after the Shodhana into the Gaumutra and Gaugrita, it is decreased to the 0.06% to 0.01% strychnine and brucine respectively. So now it can be used. Means Nux vomica before Shodhana is very toxic. After Shodhana, the strychnine and brucine were decreased to such a level that they are not that much harmful and they can be used as a drug. Okay, so this is written, the process of every drug is written into the ancient textbook. Okay, but we have to transfer this everything into our modern standardization or modern textbook. Specific dose, yes, of course, the dose is also written. The aspect of signifies the role of posology in the treatment was also there. The dose was understood as a major factor for the mild, moderate, or fast action of the drug, especially if we talk about the Shodhan dravyas like used for the Vamana or the Virechana. And the dose and type of medicine as per the type of patient was decided by the physician. Yes, at that time, the dose and the type of medicine was decided by the physician, but right now it is fixed. Okay, if we transfer into the form of tablet or like that, then it is fixed that 1 or 2 tablets are taken twice a day or like that. But at that time, the Vaidyas decided that which dose and what type of medicines are used. Okay. The another parameters are also mentioned into the ancient textbook, like the Evam Vidasya, that to be used in the prescribed manner, mode of use, to be then the Purusasya Evam, to be used in a specific type of patient. Okay, duration of use of drug means until what time you have to continue that drug, means either till the vitiated dosas are removed from the body, means 100% all the dosas will get down or pacifies, or they are settled or normally, means up to what extent you have to continue the drug. Okay, the condition of the drug on the use, not to be used in certain conditions, therapeutic contraindications were also mentioned. Okay, so therapeutic indications of the drug, if we look about that, the particular standardized drug is used in a specified disease condition, and clinical research was definitely used by the ancient researchers to ascertain the use of the certain drug in a specific manner in a particular disease condition. They know, they have a clinical knowledge of that particular drug, so they decide which drug is used for which type of patients. Formulations were disease specific, yes, of course, or they were made so by the use of a suitable vehicle. Excuse me. So at that time, the formulations were prepared by checking the prakriti of patients, okay, the requirement of patients, and say for example here Sanjeevani Vati should be used in a specific disease with a specified dose and appropriate anupana. Anupana is very important at that time. The nature of patients, right now also we check the weight and the height of patients and everything. We check whether the patient has any allergy or not, whether the patient is having diabetic or not, or like that. Every parameter of patient we also check right now also. And at that time also they have mentioned all the factors which decide the nature and quantity of the drug to be used, which is depend on the nature of patient. That all are also there. Means they check their prakruti, means if the patient is vata type of patient, means which type of dosha is prominent in that particular patient, and everything they check first. Then duration of medicine, of course, they check whether it is given for how much time. Okay, so everything was written in that ancient textbook. Method of administration, the Anupana and Sahapana are also given along with the drug adjuvant because they potentiate the therapeutic effect, the liquid medium which is serving as a carrier or a vehicle for delivering the main drug to the target organ. Say for example, if we talk about the honey or ghee, yes, they are very good vehicle for the Saptamarita Loha. Here the example is given, which has affinity for the eyes. Punarnava has affinity for kidneys. The Ashoka tends to concentrate in uterus. So this is all Anupana or the Sahapana which are given along with the drugs are very good vehicles which help the drug to absorb, which help drug to eliminate everything. Means they increase the potency also sometimes. So that is also written. Now we look to the milestones in the ASU drug standardization, means Ayurveda, Siddha and Unani drug standardization. So up to here we know that what is the ancient concept of standardization. It is always there in the textbook, but it is patient to patient vary. It is drug to drug or formulation to formulation. It's an individualistic approach. Okay, now come to the modern era. So starting from, if we look at this our Ayurvedic formulary and pharmacopoeia, what we have today. Okay, so it was started back in 1962, and these proceedings were began with the formation of the APC. And then after 2 years, the ASU drugs came under the Act in 1964 under the Section 33, Chapter 4A. Then 1969, the Ayurvedic Formulary of India, Volume 1 was published, was prepared. And then 1970, the work of standardization entrusted to the Central Council for Research in Indian Medicine and Homeopathy. Then 1978, the API published the monographs of single herbal drugs. Single drugs monograph was published first in 1978. Then 1992, the Rule 161 labeling, packing and limit of alcohol in ASU drugs came. So what was there in the Rules 161? Before that, 1992, there was not any type of rule that we have to write down each and every component of the Ayurvedic formulation on the label. Okay, we don't have to write down the percentage of alcohol in Aswas and Aristas or any formulations. But after this 1992 Rule 161, that each and every manufacturer have to write down all the ingredients along with the concentration or the percentage of that drug onto the label. If it is not possible to write down on the label, the separate literature they have to add, means give along with that bottle or whatever the container is there, in which that every detail is there, means each and every ingredients are listed over there. Okay, so this comes after 1992. Then in 1995, the Rule 168 came, and that standards of the ASU drugs as in the Ayurvedic Pharmacopoeia of India. Then in 2005, means there are lots of amendments were occurred, and by looking at the popularity of the Ayurvedic formulations and the demand in the market, they have permitted some excipients, some additives, some flavoring agents, some artificial sweetening agents, and other things into the Ayurvedic formulations, which are always into the daily intake dose in ASU drugs, and that is done into the 2008 Rule number 169. Then Schedule T came into the existence, GMP and certification mandatory in 2003. That we know that very well. Then the project was started in 2003 for development of standardization of method of preparation. Yes, this method of preparation was started in 2003, pharmacopoeia standards and self-life studies of the classical ASU drugs. Then November 2005, Rules 161B, self-life notification for ASU drugs amended against the 2010, and again amended in 2016 with the latest notification on the shelf life. So you have to also mention the shelf life of the Ayurvedic formulation on the label. Okay, you have to check it, you have to monitor it, means shelf life study that you have to compile at your formulation level, and then you have to write down the expiry date on the label. Then in 2006, rule 163A, pharmacopoeia lab set up for the ASU drugs came into existence. Then 2007, API Part 2, Volume 1 published with the this standardization method of preparation and standards for compound formulations. Then 2008 and 9, we know that each and everything that we have. In 2013, the good cultivation practice, good clinical practice guidelines for the ASU drugs issued by Department of Ayush. And right now what we have with us. Okay, so if we see the standardization of this ASU drug development and standardization and quality parameters as per this Ayush department. So first thing which required is standardization of raw drugs with the chemical profile. Means first of all, the each and every raw material, each and every drug, plant material which is used as a raw material, plant, even minerals and animal product, each and every that you have to first standardize it as per the Ayurvedic Pharmacopoeia of India. Then SOP of extract, if extract is required, but particularly in Ayurvedic formulation, extracts are not that. The herbal product, yes, of course, their extracts are used. Then the standardization of extracts that you have to do. You have to standardize your extract, then SOP for formulation, then standardization of formulation and finished product, then biological screening. If targeted biological activity is there, then safety and toxicity studies are required, and then preservation, packing, and storage. Okay, so if we look look into the standardization of single plant material. So first of all, that raw material or a single plant formulation if available, lots of formulations are there having a single plant material. Then raw material that you have to check for its botanical description, which part it is used, whether leaf, root, seed, fruit, flower, whatever it is. You have to check the botanical origin, distribution, everything. Then adulteration, you have to check whether they are adulterated or not. The season of collection, of course, you have to check. As we have seen just few slides back, that the collection time is very important because it contains, it may vary the secondary metabolites. So that's why the season of collection is very important. Then site of collection and stage of plant. Yes, it is also important. You have to check, you have to write down each and everything. Then substitution of exhausted products and foreign matter. Okay, so the second thing you have to check into the single plant material is the color, order, taste, structure, texture, means description. Everything you have to note down. Then identification by morphological, microscopical, powder microscopy, as well as the TS and everything you have to check. Then color test, chemical test by different identification tests for alkaloid or glycoside or like saponins or flavonoids and tannins and everything you have to check. Then identify by using the TLC and HPTLC. Some physicochemical parameters need to be checked, like loss on drying, means water content, moisture content at 105 degrees Celsius. Excuse me. Total ash, acid insoluble ash, total solids, pH, volatile oil, alcohol soluble extractive value, and water soluble extractive value you have to check. Then assay for the constituents like major compounds. If marker compound is there, you have to check for the percentage of marker. If not there, or if it is alkaloid containing drug, you have to check total alkaloids, total flavonoids, total saponin compounds. Then biological activity evaluations, bitterness values, astringency, the swelling factors, foaming index, the hemolytic index. These are all the assay constituents which whatever the applicable to that particular plant, you have to apply it. Particle size distribution. For powder drug, raw material, you have to check bulk density, tap density, Car's index, Horsner's ratio, and everything. So they can be applicable to the filling machine, means a hopper. If we are using this hopper and everything, the particle size distribution also, it is very helpful in case of that. Okay. Even the efficacy of drug is also depend on the particle size. The absorption and the resolution may also affected by the size of particles. Then taste of heavy metals. Yes, here we have to check this heavy metal and toxic metals as per WHO and FDA permissible limits. The lead must be less than 10 ppm. The mercury must be less than 0.3 ppm, cadmium 0.3 ppm, and arsenic 10 ppm. So we have to always check for this, and we lack whatever we are exporting outside the India. We are always lacking in this particular point, or because of so many reasons, our land is also polluted, and whatever the farmers are using some pesticides and everything. So we are always having some heavy metals and pesticide residues into the raw material. So this is what we have to always keep in our mind, that is the heavy metals and pesticides. Then microbial contaminations are need to be checked, like total viable aerobic counts, total enterobacteriaceae, total fungal count. Some specific pathogens are also checked, like E. coli presence, Salmonella, S. aureus, and Pseudomonas. Pesticide residues like organochlorine pesticides, DDT, HCH, the endosulfans, the permissible limits are given over here. Okay, pyrethroids and parathion, and all these are always under the limit, permissible limit. Then aflatoxin, yes, this is again very dangerous thing, that aflatoxins B1, B2, G1, G2, permissible limit is 0.5 ppm, 0.1 ppm, 0.5 ppm, 0.1 ppm. Aflatoxin is not added, not sprayed, but it is the indication of there is something microbial growth, there is one fungal growth, because it's a fungal toxin. Okay, so it indicates indirectly that there may be something wrong in the raw material or a product, finished product. So this is all about the standardization of the raw material, a single drug. Now we talk about the standardized operative procedures for the finished product, or the standardization of the Ayurvedic formulations. So all standardized ingredients, as we have seen, everything means 11 or 12 points what I have discussed or the point out. We'll see later in a detail also, each and everything, if time permits. Then all standardized ingredients well taken, then mixed. If the minerals are there as a raw material, they are prepared into the bhasmas, they are mixed as per the formulations. We'll see the procedure of each and every formulation later as per the Ayurvedic preparation, like the avleha, then asavas, arista, churna, kwath, and everything. And then following the guidelines in the Ayurvedic formulary of India, part 1 and 2, we have to prepare and we have to standardize them. Okay, so we see one by one each and every Ayurvedic formulation. I have not covered every formulations, but the major the very widely used formulations like churna, asavas, gritas, and avlehas, and what are also very important for point of view like syllabus of Sem 6 student. So I have included that much only, not all the formulations I have included. So if we look into the Ayurvedic formulations, they are classified into major 3 classes: solid, liquid, and semi-solid. So solid formulations are, actually there are lots of formulations, but I have included only few of them. Kwath churna, bhasma, guttika, and vatika. Semi-solid formulations like lehya, avleha, pak, pras, and grita. Grita are semi-solid at room temperature, and they are melt on heating, so they are in a liquid form. In a liquid formulations, the ark, asava, arista, and tela are there. Anjana, parpati, and all that things are there, kajali and everything, but I have not included all other formulations. So we'll see one by one, churna and kwath churna. So here I would like to explain what is the difference between churna and kwath churna. First thing, definition of both are the same actually. They are the fine powder of the drug or a drugs. Means churna and kwath churna are nothing but the mixture of drugs or maybe a single drug. Okay, so if amla churna is there, the only amla is there. If triphala churna is there, it's a mixture of 3 pala, amla, beda, and herde. Okay, but in case of churna and kwath churna, what is difference? So difference in manufacturing, there is no difference, but the difference is mainly, we can say the compounding is the difference. Means churna is taken as such with the water, While Quath Churna is always, first of all, it is prepared in the Quath, means Kasaya first, and then Kasaya is taken. Not the whole Churna is taken. Okay. So Churna and Quath Churna have this difference. Nothing much difference only. Okay. So Quath Churna is always prepared Kasaya first and then it is taken. While Churna is directly mixed with water or without mixed with water, it is directly taken with the Anupana. Whatever Anupana is there. Not always water is Anupana. The method of preparation means you have to first collect the drug which are mentioned in the pharmacopoeia, then clean and then dry, then fine powder separately, sieve them separately, weigh them separately, and then mix. Okay, so this is very important in case of Churna and Quath Churna. Each and every drug you have to powder separately. Okay, if metals and minerals are used, you have to prepare Bhasma, Sindura first and then you have to mix in them. If the hygroscopic drugs are there, if the Kshar, salt and sugars are there, you have to always keep into the closed container. You have to avoid manufacturing rainy season. This is very important. Okay. The churnas are fine powder. They are at least 80 mesh in a sieve. They retain the potency in the 1 year. Okay. They are in a powder form. They do not adhere with each other. Okay. These are some example of the formulations, means churna formulations. Avipattikar churna, Trikatu, Triflaj, Modadi churna, everything. Standardization of churna include the organoleptic evaluation, microscopical evaluation, pharmaceutical parameters like bulk density, depth density, and CAS index and angle of repose, then physicochemical H value, extractive values, and pH is very important. Chemical evaluation like proximate analysis, total tannin content, total alkaloid content, total saponin contents, or even you can estimate the particular marker using chromatography, HPLC, HPLC, fingerprinting. Microbial evaluation, total viable and aerobic aflatoxin, pesticides, heavy metal. Okay. Heavy metals are mainly done by the atomic absorption spectroscopy. Vati and Guti, they are medicines prepared in the form of tablet or the pills, and they are made up of the one or more drugs of the plant or animal origin. In that, you have to first take all the plants which are mentioned into the formulary. You have to fine powder them. Minerals are converted into the bhasmas or sindura. Then, if the parad or this gandak, sulfur or mercury are there, you have to make kajali first and then put into the khalwa or mortar pestle, grind with the paste which is mentioned prescribed into the formulation and grind until all the liquid absorbs and made a semi-soft mass which can be rolled into the pills. Then you have to add the sugandravya at the end, kasturi or karpura which is there, and then you have to roll into the small pills. If the sugar and jaggeries are mentioned into the formulation, you have to make a first pack, then you have to add all prakshapan dravyas and everything, triturate well and then make a pills. Characteristic, they are always kept in the jars and used for 2 years. Okay, if the pills contain the minerals like bhasmas are there, then they can be used indefinite period of time. Okay, they retain their color, smell, taste, form for 2 years. Otherwise, if there may be some change in that, then it may be sign of deterioration. These are some example of the vati: Agnitundi vati, Marichadi vati, Lavangadi vati, Lihari vati. So what is the standardization parameter of guti or the vati? So the organoleptics are the same: size, color, shape, taste and order. Microscopical means after you have to removal of the sugar and kshar and everything, means you have to first wash, means make a powder of their vati, dissolve into the water, then filter, and whatever the residues are there on a filter paper, you have to mount in the microscope after staining and everything. Then pharmaceutical parameters like the disintegration, friability and hardness of the pills. Physicochemical evaluation, H value, extractive values and pH. Then chemical evaluation like proximate analysis, tannin content and alkaloid content, saponin content, everything. Then chromatography, HPT. Arista liquid formulations, which are very unique and different kinds of formulations only available in the Ayurvedic pharmacopoeia, we can say in Ayurveda. These are the medicinal preparations made by soaking the drugs either in a powder form or in the form of decoction in a solution of sugar or jaggery. As the case may be, the specified period of time you have to keep in during this time, it undergoes the process of fermentation, generating the alcohol, and this generated alcohol, it facilitates the extraction of the active principle contained into the drugs, and this alcohol which is generated during this procedure is served as a preservative. So this is the Asvas and Arista. The preparation of Asva Arista. First of all, in case of the Arista, you have to take the coarse powder, make a decoction, strain it, and add into the fermentation pot. In the same pot, you have to take the jaggery sugar, dissolve, boil, and filter, and add this into the fermentation pot. Add the prakshepan dravyas, add Dataki pushpa, and allow to cover the mouth of this pot with the earthen lid. Seal the lid with the clay and cloth kapad mitti layer 7 times. Then keep under the ground or heap of the podi for particular time of period. After that, remove the lid. Check the completion of the sandana, means fermentation. Then decant the fluid and fill into the bottle. The same thing is happened with the Asava. Only here, the directly decoction is not prepared. Directly, then sugar and jaggery solution. Boil, cool, and filter. Add all the powdered drug into this sugar jaggery solution, cover the lid, and all the procedure is the same. Now, what is the benefits of this fermented product? So first thing is the removal of sugar. Whatever we have added at the time of the starting of this formulation, sugar and jaggery, is converted into the alcohol and make this product more bioavailable and eliminates the effect of such gas and bloating. Alcohol gradients, yes, at the starting point, the alcohol was 0 and at the end, maximum 20% alcohol was there. So gradient during the whole procedure gives the wide range of active ingredients extracted. Then the removal of heavy metals and some pesticides by the yeast, which are added into by means of Dataki pushpa and all other natural source. So they bind with the heavy metals and pesticide residues and act as a natural cleansing system. And for this reason, the properly fermented herbal extracts can contain less toxic chemicals than the regular herbals. Okay, they convert the toxins into the pharmaceutical agents. Yes, there are some chemical compounds in the plant which are toxic, but they are converted into the pharmaceutical agents during these fermentation procedures. The higher extraction rate due to the cell rupture, active transport, and enzymatic actions, because this much time they all contact with the solvent, so it happens. The precautions are always there because it is a fermented product. So earthen vessel should not be new, which is used for the preparation of Asava Arista. Water should be boiled first. Absolute cleanliness is required. The inner surface of the pot should be fumigated with the pipi churna. Smear with the ghee before liquid poured. Okay, it should be clear without froth. It should not become a sour. And preparation has aromatic and alcoholic odor, and it can be used for indefinite period of time. These are the examples. Standardization, everything is the same, like color, odor, taste. pH is always, you have to check. Specific gravity, as it is a liquid, so you have to some new parameters are added over here. Total solid content, alcohol content, total sugar and reducing sugar content, phytochemical analysis like total tannins and phenolics and all that. Chromatographic fingerprinting, aflatoxin determination, heavy metal determination, pesticide determination. Total solids can be done by 2 methods, one with the having sugar or honey and one without sugar and honey. Okay, so I'm not going much detail in that because of time constraint. Then alcohol content also done by 2 methods. One is by gas chromatography and one is by distillation. This is distillation unit from which you can distillate the alcohol at 25 degrees Celsius, and you can read the reading from the specific gravity of that distillate, and you can see that how much ethanol contents are there. Then semi-solid preparation, avleha, like modaka, guda, khanda, rasayana, leha, lepa, all these are under the avleha. It is a semi-solid preparation of the drugs prepared with the addition of sugar, jaggery or sugar candy and boiled with the prescribed drug juice or the decoction. They are prepared. They have mainly 4 parts: kasaya or the liquids, jaggery, sugar or sugar candy, powder or pulp or some drugs, and ghee and oil, that is Snehan Dravya or the honey. Okay, so this is the method of preparation. Jaggery and sugar candy is dissolved in the liquid, strain, remove the foreign particles, then it is boiled on the moderate fire till it become a tantumay. Means you can see that આપણે ગુજરાતીમાં એક તારની ચાસણી કહીએ છીએ, તંતુમય બનાવવા દેવાનું છે. Then you have to add all the powder drug, and then ghee is added when it is hot and honey is added when it is cooled down. Okay, then it should be not hard, it should not be so thick fluid. The pulp of the drug is added, ghee or oil is there, then it should not be stick on your finger, it should be rolled between fingers. Okay, the color and smell depends on the drugs. These are some example. Preservation and storage, they should be kept in a glass or porcelain jars. It can be kept in a metal containers which does not react with, and they should be used within 1 year. Okay, Chyawanprash and Vasavleha are the example of that. Standardization are organoleptic, macroscopical, pharmaceutical, mistake. The physicochemical evaluation like ash value, extractive value, pH, viscosity, total solid content, reducing sugar, total sugar, non-reducing sugars. Chemical evaluation like proximate analysis, HPTLC, total viable count, this aflatoxin, pesticide, everything is similar. Grita preparations in which the ghee is boiled with the prescribed kasaya and kalka of the drugs according to the formula, and this process ensures the absorption of the active ingredient of the drugs used. Okay. Then method of preparation, here the composition is divided in 3 parts: dravya, kalka and sneha. Dravya means liquid which may be one or more, or kasaya, swarasa and dugdha, etc. Kalka is the fine paste of the drug, and sneha dravya means grita or tela. The grita and tela have a similar type of procedure. So you have to take a kalka is 1 part, snehan 4 part, and dravya 16 part. This is general procedure, but sometimes it is different. In some cases, it is very okay. So you have to boil the kalka and dravya are mixed together, and snehan is then added and boil on the mild fire and stirred well continuously, so that the kalka is not allowed to adhere to the vessels, and till all the snehan dravya is come out and all the moisture removed, till that you have to boil it. Okay. There are some variations are there. I'm not going much detail in that. This is a very typical procedure of grita and tela. Characteristic: grita are solidify on cooling, tela does not. It will have a color, odor and taste according to the drugs used. So grita are generally used internally, and sometimes they are also used externally, and oils are always used externally. Okay. So these are some examples. The standardization, refractive index, and the specific gravity, weight per ml, saponification value. Here they all parameters are changed because it is oil or the ghee is there. So saponification value, iodine value, acid value, peroxide value. Okay. Mineral oils, whether they are present or not. This is everything is given in the appendix number 3.12 in the Ayurvedic formulary, microbial limits and aflatoxins. The last one is the bhasma. Now we know the bhasma, that means ash or a product which is obtained by incineration or calcination of materials. Metal, minerals, some plant products as well as the oceanic products are also used as a raw material to prepare this bhasma. And when these are subjected to heat in a specific style, they are naturally converted into the different forms, and this form is known as a bhasma. Mainly 3 parts are there for preparations: the purva karma, pradhan karma and paschat karma. Purva karma, that means shodhana, purification. Pradhan karma, that means incineration and calcination, maran. And then paschat karma, that is the lohitikaran or the amritikaran. Effect of shodhana, we know that the purification is occurred into the first step, that is the shodhana. The materials become free from visible, invisible impurities. Mass of minerals converted into the fine and brittles. Development of fineness and brittleness facilities into the bhavana process. Partial reduction takes place, and reduction of the organic therapeutic properties in the inorganic material. So this is the bhavana, means trituration. Advantages that uniformity, mixing, and materials divided into the fine, and this is called the marana and bhavana. Then pelletization, you have to make a chakrikas, okay, and then dry under the sunlight. Then sarvasamputikaran means you have to make seal, keep in between 2 earthen sarava, and the seal 7 kapad mitti layer are done. Then incineration, puta system of the heating. Puta is specific system of the heating for the incineration. The depends on the nature of your metals or the materials which are going to be bhasma. You have to select this puta system, means it depends on the size of that. At that time there were not a measurement, so gajaputa, kukutaputa, varaputa, gajaputa, like that, such kind of measurement was there. Depends on the size of that animal, they have decided the size of puta. Okay. The next is the marana or the incineration. You have to make like this puta and keep in the sarvasamputa inside that, and cow dungs and everything, and you have to fire it. Then sanskaras of the bhasma, post-operative treatment. There's amritikarana, it removes the remaining blemishes of the bhasma. Then lohitikarana, develop the desired red color in case of the loha and abrak bhasma. Okay. So these are evaluation parameters like varna, color and float, vari taritva, here you can see the Varitaritva, Rekha Puritva. Okay. Then Unamana, Anjana, Sanniva, softness, Nischandritva. So this is all parameters. Chemical parameters like the tastelessness, the Nirdumatva, absence of fumes. Okay. Purva Bhava, then irreversibility, Nirudva, Amla Pariksha, sour taste is not there. Okay. So these are all the parameters that we have to check. These are the modern parameters which are checked: EDX, SEM and TEM, and this XRD, this X-ray diffraction and everything, HPLC, NMR, IR, MALDI. These are all the parameters which are checked into the BUSMA. Okay. So with this, I would like to thank everyone for listening me. And whatever I know about the Ayurvedic formulations, actually it is my limited knowledge. It is not all about the Ayurvedic formulations, but whatever I learned into my last 15 years of experience, I have shared with you. Thank you very much.
Thank you so much, madam, for the wonderful session. You have very nicely covered all the points, starting from the what is Ayurveda to the difference between the past and present scenario related to Ayurveda, then multi-drug, multi-target mode of action of the Ayurvedic medicines, and also you have covered nicely about the need of standardization by giving the certain examples of different marketed formulations, and those are explained by the different WHO parameters, and also you have explained nicely all the different Ayurvedic formulations with the standardization parameters. Thank you so much, madam.
I hope I have covered all aspects, I think.
Yes, madam. You have covered all the things, and as truly as you said that this is all in semester 6 syllabus. So the students are very much benefited by the session. And once again, thank you so much, madam, and thank you all for joining us. Thank you.
Thank you, Parul University and team, Tanvi madam and everyone. Thank you.
Thank you, madam. Welcome, madam.